Okay, we're going to officially get going in about 2 minutes. Just before we officially get going, just to remind you about your final exams. So some of you have been coming in to check over your short answers, and as you know, we have a slightly different marking scheme available for you for the final exam, where it's a little bit more open-ended. There are no right or wrong. There are better answers. There are other types of answers. You can actually write different types of constructs. But the one thing that I'd like you to make sure that you are always kind of doing is to be able to provide the promoter a really brief rationale for why you're using that promoter, and then any drugs or toxins. So for example, today you're going to learn how to kill cells specifically. If I want to eliminate a group of cells, and only that group of cells, how can I do that in a very, very specific manner? And you might ask yourself like what that might look like on a final exam. So we are going to, again, ask you for details that you should be able to provide us on the final, so please make sure that you are aware of that. We'll also be posting some other times that might be available for other tutorials. There are two groups that are kind of going right now. One group has already met twice, so they're a little bit more ahead, and the other group is just about to get started. So let me know if you are interested in that. So I'm going to go through the last part of the thymidine kinase a little bit later on. Bernice, I think the microphone's on. Okay, I'll let you do what you need to do. And then what we're going to be talking about are these different types of extracellular vesicles, why they might be of interest to you in cell and systems biology or as neuroscientists, why they might be useful in delivering different types of things. Some of you, for example, are interested in bioengineering and this idea of like barcoding cells. In fact, barcoding cells has been around for about 5 years to identify specific groups of cells that have been labeled with a specific marker. But at the end of today, you should understand that actually barcoding and the use of specific DNA markers, RNA markers, actually might not be as useful as we once thought, especially in relation to these multivesicular bodies and the way these exosomes work, and just again to make sure that you understand why these are thought to be effective. And we're going to switch gears after the break to talk a little bit about Alzheimer's disease, and Alzheimer's disease is also a topic that I'm hoping that you'll make an impact on, and then we'll sort of wrap up the course. All of today's lecture, by the way, both the first half and the second half, has some pretty good representation on the final exam. So I'll ask you to try to pay as much attention as you can as we're going through this. So I started talking to you about the fact that we need a way in which to eliminate only those dividing cells that are also migrating. Other individuals previously had used different types of drugs, things like cytosine or rabinoside or Ara-C, as well as other types of chemotherapeutic agents to eliminate dividing cells. But if you eliminate all dividing cells, that might actually impede on the immune response. It might actually impede on a number of other things, not just on the cells that are migrating to an area of stroke after stroke has occurred. And other individuals have tried delivering this HSV1 thymidine kinase using other non-specific promoters. So GFAP, for example, as we learned in the last lecture, is actually a general marker for proliferating cells. They're in the lineage of from the subventricular zone as well as in the dentate gyrus. And we know that nestin is also an earlier marker for all dividing cells and not just dividing and migrating cells. So these previous ablation techniques where specific groups of cells were being eliminated either through chemicals or through genetic methods weren't really all that precise. So then we started looking at double cordon. So double cordon is indeed a gene and a protein, and therefore a marker that's specific to dividing and then migrating cells. And so if we were to take part of the double cordon, or DCX, gene, and then we were to also express in those cells that have double cordon, also express things that might lead to their selective ablation, things like thymidine kinase, we might have a mechanism or a potential model system that we can use to eliminate this specific group of dividing cells that divide after stroke and that also migrate to the area of stroke. And this is why we're interested in double cordon. So cells will divide after stroke, and cells will migrate to the area of stroke. But what happens if we delete them? Is stroke any better or is stroke any worse? And again, when we look at a number of other types of markers, we know that double cordon will eventually form the neuronal lineage. So they will eventually, when we mark them initially with double cordon, as is shown on the second bullet point, they will eventually form things that look a lot like neurons. So they might have markers like beta-3 tubulin, which is a neuronal marker, or MAP2, which is a neuronal marker, but they don't have markers for oligodendrocytes like lactocerebroside or GFAP, which is also an astrocytic marker. So again, if we look at the way that in which they did this, they were able to induce the expression of double cordon thymidine kinase in specific areas where we already know that proliferation as well as the generation of these neuronal precursors occurs, which is the subventricular zone as well as the dentate gyrus. So we get those cells in that particular area. Everything else in these different mice looked perfectly normal, so they didn't look any different in terms of reproduction, size, etc. So now we need to understand a little bit about how thymidine kinase could potentially work. And this goes back to what I was telling you just before we got started in terms of being able to provide some of the details. So thymidine kinase is not an endogenous kinase. We have to introduce it. It's not normally found in cells. It's found in cells if they get infected with herpes simplex virus, but herpes simplex virus, again, is not going to normally be part of the repertoire that we would find within the brain. So why is thymidine kinase potentially bad? So one of the things that we know can happen is that if we were to somehow deliver virally this thymidine kinase or transgenically develop these thymidine kinases, which is normally part of herpes simplex virus or HSV, then in response to this drug, and again, I've also asked you to make sure that you know how these different things work, the drug or toxin, in this case, ganciclovir. So if we were to give an antiviral drug like ganciclovir and the cell happened to express thymidine kinase, the first thing that would happen is that ganciclovir that's normally not found would enter into the cell, and only in the cells that are expressing thymidine kinase, and only those cells, it will actually get phosphorylated. And that first phosphorylation step is key. Normally we don't think about phosphorylation as a way to trap molecules, but once that cell has had ganciclovir freely crossing the membrane because it is lipid permeable, there is no way for that cell, for that molecule that's phosphorylated here at, on the left to actually leave the cell. It is trapped intracellularly. And once it is trapped intracellularly by that thymidine kinase, that first step, this ganciclovir will then, through other intracellular and endogenous kinases, now be phosphorylated on a different region and have a diphosphate, and then eventually those kinases will allow for a triphosphate molecule of ganciclovir to occur. Why all of this is really important is in the cells, and only in the cells that are overexpressing this thymidine kinase, this triphosphate form of ganciclovir actually is an purine analog, so it looks a lot like a base pair that is normally found. And in the presence of this purine analog, this will actually impair the DNA polymerase, it will inhibit it, it will eventually stop all cell division within that group of cells, and eventually it becomes toxic. So any cell, in other words, that is overexpressing thymidine kinase and is exposed to ganciclovir undergoes this process, and it then causes that cell to die. So this is how we have a specific way to ablate or lesion this group of cells. As long as we are delivering it to one group of cells, and we are through the use of double cordon, then we know that this is a specific way in which to target and eliminate these cells. Again, in the neuronal system, we don't normally have this bystander effect where this toxin can actually spread from cell to cell. In other systems that you might look at later, either in cancer or in terms of cell and systems biology, if the cells are capable of forming gap junctions, this triphosphate analog will eventually go and impede the activity of all these cells. But within the nervous system, where these gap junctions don't exist, the cells that are specifically targeted with thymidine kinase to overexpress it will be the only ones that will actually die
in the presence of ganciclovir. So here's a way for us to have a transgene specifically targeting a group of cells, and then to use a toxin to be able to kill off those cells. So this is the second method that we looked at. We have also looked at the inducible diphtherotoxin receptor system to be able to do the same types of things in a slightly different way. So again, there is no need to memorize these structures. You're not going to be given any structure diagrams on the final. You are not going to be asked to draw any of these structures out. The only thing you should take away from this slide is the method by which thymidine kinase works, and you should be thinking about how you might apply it on the final exam, because it's likely that you are going to be asked to apply something that we learned on the final exam. The next series of slides, again, are just to give you a little bit of context for what's happening. So when we go in and if we were to expose a wild type cell over here on the left in panel D, and we were to look at ganciclovir and inject ganciclovir into these animals, no thymidine kinase, they're just wild type. In fact, we won't see any effect on proliferation, which is what we're picking up after the introduction of BRDU. So we see a lot of dividing cells in the subventricular zone, just as we would normally expect. Ganciclovir has not targeted them. They don't express thymidine kinase, and these little brown dots that you see here represent the dividing cells that are BRDU positive. So we've injected them with BRDU, we allow them to divide, then we use an antibody to stain for BRDU, and ganciclovir has done nothing to them. If we were to go into the double-quartan thymidine kinase mice, and then we were to do exactly the same thing, inject them with BRDU to see dividing cells, they take up BRDU, and then we can detect that BRDU with an antibody, and then we were to expose them to ganciclovir because they express thymidine kinase, we will actually see that we don't see staining. There is no staining because there are no dividing cells. We have specifically eliminated them. One of the things that is always a concern in these types of studies, if you are killing so many cells, would you not expect to see a compensatory immune response? Should we not see more microglial cells or invasion of macrophages? And the middle panels that you are seeing here try to address whether or not this occurs. And again, just to make sure that we're not spending a large amount of time on this, the bottom line is that in the presence of ganciclovir and in the presence of selective lesioning of these different cell types, in fact, we don't see the activation of microglial cells. We don't see more microglial staining. So when we use an antibody like OX42 that stains for microglial cells, we don't see more microglia. We do not see activation of microglial cells following the elimination of the double-quartan thymidine kinase expressing cells. So we don't see an immune upregulation, so also a good model system because, again, the microglial cells themselves might be confounding our results later because we know after stroke has occurred, there is usually microglial upregulation. So IBA1 is something that we talked about previously as well, which is also a microglial marker. So IBA1 and OX42 are both showing exactly the same thing, that after we kill these cells, there is no compensatory upregulation of the immune response. We do not see more microglial cells after we deliver ganciclovir. So again, going back to what these authors originally wanted to do in this PNAS study was to use focal cerebral ischemia. So they wanted to do this using middle cerebral artery occlusion, or the MCAO model. Again, another good idea for you to make sure that you are aware of, because you will get both multiple choice test questions as well as short answer questions potentially, is to know the function of the different types of model systems that we can use to induce stroke. So we talked about different model systems like endothelin-1, which causes vasoconstriction. We talked about other model systems like MCAO, and we talked about, again, how these different model systems can be specifically used in this context. So here, in these double Corton thymidine kinase mice, we do this middle cerebral artery occlusion, and then after we do this occlusion, after we have induced stroke, then we will actually wait and then deliver ganciclovir to kill off the dividing cells. So we normally see division after stroke has occurred, and we want to know, after stroke, what is the purpose of those dividing cells? It happens naturally, and if we eliminate that, does it make it better or does it make the stroke worse? Again, if you were to continue on with this ganciclovir application, you do also some sensory motor testing, and then you analyze the brain after sacrificing these mice. And I'm going to go through some of these different behavioral tests, ones that I'd like you to be familiar with as a way to test for sensory motor control, and if you have better or worse sensory motor control. And the ones that we're going to be talking about are the rotor rod, limb placing, and elevated body swing tests that we're going to be talking about. So again, we normally see this ischemia-induced proliferation of these precursors in areas throughout the brain, but especially within the subventricular zone, and then they migrate to the areas where stroke has actually occurred. In this particular model, the way that they set up the middle cerebral artery occlusion, and I told you you did not have to know about the different branches of the middle cerebral artery. One area of the brain that is affected by this is known as the striatum. The striatum is really important for motor control and motor function, so it makes sense that if you are causing ischemia in the striatum, you would expect to see a deficit in motor function as well as motor activity. So again, ipsilateral meaning on the same side as the actual stroke has occurred. So there's ipsilateral, same side as the stroke. And we want to know what will happen if we were to eliminate these dividing and migrating cells. Will the stroke be any better, or will it be worse? So we know, for example, in the ganciclovir treated mice that contain thymidine kinase delivered specifically through double Corton to the migrating proliferating groups of cells, that if we eliminate them, that we indeed see no migration, no double Corton staining at the level of the striatum. So normally we would see some staining there, but now in the presence of ganciclovir, we have eliminated them. So our model is still holding up. And again, this neurogenesis and neuromigration is what we're trying to look at. So if we were to take a look, this is the standard rotor rod test. So the animal is put onto this revolving wheel, and then we just want to know how long can they stay on this rotating wheel or this rotor rod. If it has perfect motor function, then it will stay on longer. And if its motor function is impaired after stroke, it will fall off sooner. So we're just measuring the time, how long it stays on, or how quickly it falls off of this rotating wheel. And again, we normally set it so that there's a baseline value, and we want to see how long it can actually stay on for. So the time on the rotor rod is going to be much worse. The animals, after we eliminate the migrating and dividing cells after stroke, it cannot stay on as long. So functionally, behaviorally, we see that the migrating cells are probably making a difference. They are probably helping in the recovery of the animal 14 days after stroke has occurred. Again, the same types of things in terms of placing. It's not shown for you here, but if we are causing a stroke and the loss of neurons on one side of the body, on the other side of the body, on the contralateral arm or the limb, we would expect to see some sort of deficit. And what we're looking at is what is called a placing score. So if you were to see an animal, like a mouse, it normally stands up on its hind paws, and as it's exploring its cage, it will use its left and right arms, and it will kind of touch the glass as it's standing up and explore the environment around it. And we can actually measure, is it the same for both arms, or is it actually the placing score, is it more for one side or more for the other. And when we lesion specifically and have a stroke on one side, and then we eliminate the proliferation as well as the migration, in fact, we see a deficit. It is using one arm much more than it is the other. It's not sort of equally balanced out, and this placing score itself is actually lower. If we were to suspend an animal by its tail after causing a lesion of the areas that's responsible for motor activity like the striatum, it tends to swing equally in both directions, to the left or to the right. If we were to specifically lesion one side, we would actually see a deficit in the way that it swings. And this left body swing, if it's swinging to the left or to the right more, we actually see a deficit in its left body swing. All of these different data suggest behaviorally, through a way that we can measure, in these mice, after we have eliminated the proliferation and then the migration to the area of stroke, that by doing so, we actually affect performance. None of these mice, after ganciclovir, actually perform all that well in terms of motor tasks. They don't perform well on the rotor rod, they don't do well on the placing test, nor do they do well sort of on how they swing their body to the left or to the right. So all of these suggest that there is a big deficit that we can identify. In terms of the biochemistry, so functionally, we see that eliminating this group of cells results in a behavioral deficit. It looks like that the neurons that are supposed to migrate to the area of stroke do indeed enhance recovery 14 days after stroke has occurred. So biochemically, if you were to look at, is there more cell death or is there a greater volume of stroke and loss of cells, can we identify whether or not this occurs? Again, this is a slide that you probably want to go back and review prior to the final exam. And one of the ways that we can do this is to use a compound known as TTC. And this tetrazolium salt is a way for us to examine the function of the mitochondria within neurons. And we know that mitochondria play a big role in this. You don't have to know the long form name of what TTC is. And we can see what is the overall size of the stroke volume after we have eliminated the migrating cells. And if we give ganciclovir in the presence of this thymidine kinase, will the size of the infarct, will the size of the stroke look bigger or will it be smaller? And in fact, this increase is actually due, we do see an increase in the infarct volume, which is shown here. So in red, when we apply to a slice, so this is a slice that we would take out of the brain, and while the slice is still alive and it is being perfused, we can actually apply TTC. And TTC is a white salt, and it is also a substrate of a lot of different cellular dehydrogenases, and it's also part of that mitochondrial chain. And one of the things that happens, if the mitochondria and if the cellular dehydrogenases are intact, this white salt actually gets turned into a red color, which is what you're seeing. So red actually represents here living viable cells. So what we see is that we get a stroke, we get the loss of all of these neurons, they can no longer metabolize TTC, and it stays as a white salt, and all of this other part of the brain, because we only do the occlusion on one side, and so it's limited to this one side, ipsilateral, the dye is metabolized to this reddish color. So if we were to look at an animal that had the same type of stroke and then had all of the migrating and dividing cells eliminated with ganciclovir addition, so the thymidine kinase takes and phosphorylates ganciclovir, it gets trapped in the cell, and then it eventually causes the loss of the cells that it's expressed in, we see that the stroke volume is actually much bigger. So there's more cell death, or there are fewer remaining cells after ganciclovir addition. So the infarct size does actually increase in the presence of ganciclovir, meaning that not only behaviorally in the last few slides that we looked at, but also in terms of the histology, when we look at it, or the biochemistry, we actually see that these migrating cells and these proliferating cells make a big contribution to the recovery after stroke. So again, if we have no migration and no neurogenesis, the bottom line again being that we will have a bigger infarct size, and they're just confirming that in this series of cells, in this series of panels, which again, you don't need to worry all about. There's nothing that you should try to be memorizing or anything else. It's just to show you that in terms of the double cortin and thymidine kinase, that overall we don't get this migration, we don't get this neurogenesis, and especially on the side that is treated with ganciclovir, we don't see the type of recovery that we normally would after we induce this stroke. So that sort of in a nutshell is a way for scientists to have done a very specific lesion to only the dividing and then migrating cells, only the cells that overexpress double cortin, and only the cells that have thymidine kinase. Even if we have all of that, it doesn't matter until we provide the toxin that is going to activate it. And again, without ganciclovir, these mice would look no different. And it's only after ganciclovir that we see these behavioral and biochemical changes that we've just gone through in the last few slides. So again, I'm going to also indicate to you that this section is also going to be important on the final exam. I'm hoping most of it is a review for you. And a few years back, people started to question whether or not we should be doing these types of transplantation of stem cells into an area after a stroke. And it was very controversial because, again, we talked about Gordie Howe leaving Canada, going down to the Caribbean to have his procedure done, where stem cells were actually injected into his brain. He had really good recovery, but we really don't know what that recovery actually is a result of. And a lot of scientists believe that stem cells actually shed these types of extracellular vesicles, and it's not really that the stem cells are replacing cells, but they're releasing compounds in these vesicles that actually might be of benefit to the damaged brain. So this has sort of changed the way that we might be thinking about how we deal with stroke and how we might be enhancing long-term functional recovery. So if we don't have to inject in stem cells, maybe all we have to do is to inject in these extracellular vesicles. So up until now, what we've been trying to do is to reduce reperfusion injury. So drugs like TPA, for example, help to reperfuse tissue, and we want to reduce sort of the red infarcts that might result. We want to get fast recovery, and we want to make sure that the vasculature and the blood vessels are recovering well. But we don't really have a good regenerative therapy. So we fixed blood flow back to the brain. There are no issues with the blood vessels, but we've lost a lot of neurons. How do we help those neurons to recover or to regenerate afterwards? And again, except for what we know about tissue plasminogen activator and its health and recovery, and except for potentially stem cells, we don't really have a lot of good therapeutic approaches. So one way that we might get around this is to, again, look at papers like the one that I just showed you, and you don't need to worry about memorizing specific details of specific papers or any of the titles, etc. But one of this is to use what is collectively called extracellular vesicles. I'm hoping that you've at least encountered them before in your first or second year biology class, because increasingly they are a really big part of biology. So these extracellular vesicles, which I'm going to abbreviate as EVs, are these membrane-shed vesicles of different sizes. So we have microvesicles that are anywhere from 50 to 1,000 nanometers, or we have exosomes, which are generally smaller at about 40 to 150 nanometers in terms of their overall size. These get produced by all the different cells. We now know in 2018 that all cells, oligodendrocytes, astrocytes, microglial cells, neurons themselves, they are always producing and shedding these extracellular vesicles. So we're now very interested in understanding that. We have so much work to do to catch up to what these extracellular vesicles are, that we're trying to figure out what they're really doing. So what are they doing there in the first place? Why do they need to be shedding this? Are they actually communicating different things through what they are shedding? So one of the reasons why we're really interested in this, if you give someone stem cells, that's it. We have to wait. We have to wait sometimes a year or 2 years to see if there's any effect. We have to limit ourselves in terms of the dosage. And there really isn't a prescribed way in which we can deliver like stem cells 3 or 4 times. With these extracellular vesicles though, we can actually deliver multiple doses. It's more like a drug than stem cells. Stem cells, we don't really know what's happening. But with these extracellular vesicles, there is more of a likelihood that we can give it once, we can wait a week to see if there's recovery, we can increase the dosage and then deliver them again. We can also store these extracellular vesicles much more readily and much more easily than we can stem cells. Stem cells, to hold them in a certain state or to hold them at a certain number actually requires a lot of time as well as effort. You need a lot of specialized equipment that most people are not able to have. And in addition, how are you going to stereotactically open up someone's brain and then drill through and have precise stereotactic coordinates to be able to deliver those stem cells to the area that is required? So this advanced medical training, also we don't need to do it. In theory, if we have these extracellular vesicles, we deliver them through a blood vessel, through a standard medical procedure like IV injection, they'll get to your brain and they may be able to do just as good a job as stem cell delivery. So a lot of previous research has actually suggested that this might actually be beneficial. But there's a caveat. We're really far behind. We don't really understand the way in which this works. We can identify them. We know they exist. We know that they're happening all the time. We know they change after stroke, but we really don't know much more than that. So previous stroke studies had actually identified that one of the best ways to repair the brain is not to actually use extracellular vesicles from neuronal tissue, it's to actually derive them from what is known as mesenchymal stem cell tissue. So these mesenchymal stem cells, again, if you're taking other CSP courses, have been actually found to be better at brain repair than stem cells that are from the neuronal lineage. Doesn't make a lot of sense. It's like telling someone that if you go to U of T undergrad, you're more likely to get into U of T meds than someone who doesn't go to U of T. And we know that that doesn't happen, right? Because there's a bit of a disconnect there. So we know that these mesenchymal stem cells, even though they're not part of the normal lineage, actually perform better than the neuronal stem cells. And this might be because the extracellular vesicle cargoes that are being released. So I told you that all cells within the CNS, every last one of them that we've been able to identify, they are always releasing these extracellular vesicles. They are carrying cargo. They are carrying proteins, RNA, DNA, etc. And these are actually really important. And these biological properties are ones that we need to be able to identify. And sadly, we only started to do this in the last 5 years. There's a great deal of work for us to be able to catch up on. So people wanted to start to identify, is it really the cargo from these different stem cells that is important? So when you look at the extracellular vesicles that are derived from either mesenchymal stem cells or neural stem cells, is there a difference in the cargo that they deliver, and therefore is there a difference in the functional recovery following stroke? So again, just as a reminder to you, these mesenchymal stem cells are again multipotent adult stem cells. They're found in a number of different tissues, so they exist in the adult stage. So they're multipotent, found in the umbilical cord. We can also find these multipotent mesenchymal stem cells in the bone marrow as well as in adipose tissue. We can reprogram them again from the adult stage as well. They are self-renewing, they divide, they can differentiate into a number of different tissues, but just as I told you earlier, stem cells normally favor the type of cells that they exist in whatever niche they're found in. So neural stem cells will eventually form cells that are found in the brain because they're neural stem cells. So in the same way, if you were to find a mesenchymal stem cell, they favor forming things like bone, cartilage, as well as adipose tissue or connective tissue. But even though they're forming a different cell type, they might actually be beneficial following stroke, or in cases of neurodegeneration and chronic loss of cells. And again, there's not a lot of research done on Alzheimer's disease or Parkinson's yet on the role of mesenchymal stem cells and their extracellular vesicles as well. In terms of this bystander effect, again, we don't really understand how this works. It's bizarre. So if you were to GFP label mesenchymal stem cells, and you were, for example, to induce heart injury, so you might think that the heart might benefit the most, and actually it turns out that it does. If you were to inject in GFP labeled mesenchymal stem cells, induce loss of heart muscle tissue, and you were to go back and stain the heart after mesenchymal stem cell incorporation, you would think that you would get GFP labeled cells in the heart, but in fact we don't see them there. In fact, we don't see them anywhere. They disappear. And yet they have a beneficial effect, and we call this sort of a different biological bystander effect. We don't really know where they've gone, but they have been a benefit to the system that has been damaged. So again, they will also migrate into areas of injured sites. They have this homing property. They normally promote functional recovery, and they are also known to modulate immune responses. We also know, for example, that if we were to deliver this intra-arterially, these mesenchymal stem cells have been really useful in animal models of stroke. Not neural stem cells, but mesenchymal stem cells. And if we were to deliver them directly into the brain through the intracerebral route, you'd have to inject them directly into the brain, open up the skull, go through the meninges, etc. It has also been shown to be really successful after clearing out the hematoma or the blood clot that was associated with it. So a lot of different research has suggested that these might be really, really useful. So again, there are a couple of things that I want you to know from this slide. You'll probably see them on the multiple choice. You'll probably see them as part of a short answer, or you might incorporate this as part of your short answer. I don't need you to memorize this entire list of different molecules that are found on here, but we do see different types of molecules that are associated with these different types of extracellular vesicles. All of these are extracellular vesicles. Exosomes are extracellular vesicles, microvesicles, and then these apoptotic bodies that I'll talk about very briefly as well. Again, this slide here is just to give you an overview of the potential ranges in size. So even though microvesicles, they can sound like they're the smallest, in fact, they're much bigger than the exosomes that I'm going to be talking to you about, and they're roughly on the same order of magnitude in terms of size as these apoptotic bodies. So again, if we're taking a look, if we were to look at why these extracellular vesicles might be important, it turns out that they have a number of genetic cargo. They are carrying different types of molecules. They are also useful for transferring receptors from one system to another. That was not thought to happen. It's only, again, sort of in the last 5 years-ish that we understand what's going on. And maybe they might be really useful for initiating different pathways that would normally have been thought to be impossible without the delivery of DNA or RNA or microRNAs, as well as the different receptor types. So by doing so, by transferring receptors, by transferring genetic cargo, they might be able to modify cell fate, they might be able to modify cell function, and they might be able to modulate synaptic plasticity. Again, we're just now sort of understanding what these different types of things might be doing. So this slide is one that you might want to circle if you haven't seen it in a while. And these cell-free strategies means that we don't need cells, we just need these extracellular vesicles. So whether these are microvesicles that we are delivering, or we are talking about exosomes, or we're talking about eliminating things like apoptotic bodies, these are new cell-free strategies to controlling stroke. And we think one of the ways in which we might be able to do so are through these different exosomes that seem to be incredibly powerful. And one of the reasons why we think they're really important is because they contain a lot of these microRNAs. And many of you will know that these exosomes, as part of these that contain these microRNAs can affect
the transcription of DNA in neighboring cells. So one of the processes that we know and that you should be familiar with is that exosomes begin through the endocytic process, and these early endosomes that are created from the cell surface will then package in a number of different exosomal proteins. They will also pack in cytosolic proteins from this cell and could potentially transfer it. So for example, we know AMPA receptors are in these early endosomes, so you could theoretically transfer AMPA receptors from one cell type to another. And they also contain these microRNAs. And from these early endosomes, they can then bud off into what are known as multivesicular bodies. And some of these multivesicular bodies can then be trafficked back up to the cell membrane and then released in the form of these different exosomes. Again, all different sizes. They can range in size quite a lot. These microvesicles are different. They do not require endocytosis to begin with. There is no endocytic machinery that is directly implicated in microvesicle formation. So normally things that are found on the cell surface, and again, receptors are found on the cell surface, might be really important. We could also package in cytosolic proteins or cytosolic messengers, and then they can be packaged and shed in terms of these larger microvesicles. And many of you will know that apoptotic bodies are generated through this blebbing process as cells die, and they are releasing this genomic DNA that could theoretically be taken up by other nearby cells. And this is the way that this genetic transfer might directly affect cell fate. The ones that I'm concentrating on are these exosomes, but if you're looking at these microvesicles, we know that they contain and can package a lot more. So not only microRNA, but also messenger RNA directly, as well as DNA. So they are different in terms of the cargo, what they're carrying, and potentially how they can alter the function of their nearby neighbors. So this is a good sort of slide to know in terms of how we separate out, how we distinguish these different types of cells. So ultimately, we think that these extracellular vesicles that have these heterogeneous populations that have differences in size and what is packed into them might actually be really important because they will package different types of proteins, bioactive lipids, the nucleic acids that I've just gone through with you, and these will then be transferred to the neighboring cells. This might be a new way that we hadn't thought of before of cell-cell communication. This might alter the activity of these target cells as well. We know that there's a lot going on outside the normal synaptic transmission. So this is the reason why neuroscientists are particularly interested in this. We used to think the only way that neurons communicated with each other was through direct synaptic neurotransmitter release, receptors taking up the signal, and then repeating itself on and on, onwards from that. But we now know that in the CNS, there's a lot of communication between neurons and glia, between glia and other types of glial cells, and this is important under normal physiological conditions as well. One of the first papers that came out in 2007, so just over 10 years ago when people started to get interested in this, is that it might actually play a really big role in synaptic plasticity. If you can alter extracellular vesicle release, you can actually alter and reduce the amount of plasticity that is found between synapses. So we know that this is an important sort of biological process. And again, this is just a slide for your own information. When we look at the complexity now of all of the different signaling that goes on, not just in between the different cell types, so between the different types of glial cells, between the glial cells and the neurons, and between all of the other different glial cell types, we also know that things are coming in directly from different cells found within the blood, whether these are mesenchymal stem cells or other types of cells. This has become a much more complicated, but much more exciting process. Because if we can understand this sort of new pathway, we might have some new avenues to be able to treat things like stroke, or maybe even things like Alzheimer's disease or neurodegenerative conditions that we thought would be impossible to be able to treat. So we do think that these extracellular vesicles will actually have a tremendous impact therapeutically on the brain. We just haven't really figured out the best way to be able to do this.
So I think at this time, if it's okay with you, we'll take a little bit of a break now. So we'll come back in about 10 minutes, so just after 2 o'clock on this clock here. And then we'll just pick up with the rest of the lecture. Again, there's some more material from the second half that you need to know for the final exam as well. So I will encourage you to take down notes as we go through it. So let's take a 10-minute break from now and then come back and pick things up after the break.
Okay, we're almost there, almost at the end. So the next sort of 50 minutes-ish, I guess, and this material is on your final exam, so there are things for you to think a little bit about. I'll point out what you need to know. Most of it is not stuff that you need to know. Many of you will have heard of Alzheimer's disease, and I'm pretty sure that you're familiar with Alzheimer's disease, and that part of it, great. I want you to make sure that you know it and why it's impactful and why we're worried about it. But I want you to actually know the cell molecular biology and the impact that it has on the synapse rather than sort of the more global social impact that Alzheimer's disease might have. It is something that's in the news an awful lot. It's increasingly in the news for a variety of different reasons. And again, as we started off this course, at the very sort of 5.5-week point many weeks ago, I wanted you to understand that neuroscience and neurobiology should always have some sort of impact on your lives. So I'm hoping that you'll take a little bit away from this particular lecture. So one of the reasons why I'm particularly interested in it, so when I left undergrad, and actually had no idea what I was going to do next, by a really bad series of coincidences, I ended up here, which is now sort of a different building altogether. But when I was doing my grad work, this used to be the Center for Research in Neurodegenerative Diseases, and this is where I started my grad work. And my office was actually right here, right above the entranceway, and it was one of those things that I really hadn't given any thought to. So in undergrad, it was sort of a blur. I just took a whole bunch of courses because I needed it for my physiology major, and I really wasn't sure what I was going to do with my life. I didn't really want to go to grad school, but then again, I also didn't want to be a bank teller. So I decided that, hey, and there's nothing wrong with being a bank teller, by the way. If I could go back, I might actually take that job versus where I'm at now. I went to grad school, and I had no idea what grad school was going to be. But I did know that the only part of neuroscience that I kind of liked, because the whole thing about saltatory action potentials, things about channels and voltage gatedness really didn't excite me, but the aspect of neurodegeneration and knowing a little bit about the processes involved in how cells die, that was really interesting. And I didn't really give a lot of thought to it, because again, grad school was almost like undergrad in a sort of different way. So you go through, you take your courses, you don't really think about what you're doing. And grad school is almost the same thing, to be quite honest. Grad school is where you show up, you do your lab experiments, you kind of head toward your goal of finishing your thesis, and then you're done. And then you don't really get a lot of time to think about it. And so I was working on Parkinson's disease, I was looking at different models for Parkinson's, I was looking at apoptosis, and I was just going through the motions of understanding these different types of things, and it had no personal meaning to me at all, period. Because one of the things that happens in grad school is that, just like you do now in undergrad, you kind of focus on your studies, and you get very insulated in terms of focusing on what you think is really all that important. And why I'm mentioning this is because this individual on the left. So how many of you know who he is? That's great, because if you knew who he was, I would actually be really freaked out, because that's my granddad. And my granddad is someone that I didn't actually know a lot about him. So he was sort of in Brazil. I didn't really see him very much because we were up here in Canada. And then my dad started to worry about his father many years ago. And I didn't realize at the time that actually a lot of the stuff that I was studying was actually directly related to what he was going through, because he actually had a very bad form of Alzheimer's disease and dementia. So by the time he passed away, so he had a number of different complications, completely lost his memory, lost his emotional control, had the worst form of Alzheimer's, which was like bedwetting, just complete loss of his sympathetic and autonomic nervous system, and he was like completely beyond what I sort of knew. And again, that sort of happened. I was in grad school. I remember hearing that he passed away. I remember hearing that maybe he passed away through Alzheimer's disease. I made a little checkbox. Okay, you know, I'm sad to hear that my granddad passed away, but I didn't really think a lot about it, even then. And then I started to hear that my uncle passed away from Alzheimer's disease. Then I started to hear that my cousins passed away from Alzheimer's disease. And then I started to pay more attention, because then it became more of a, not just a research-oriented thing, but a very personal thing. The other reason that I'm putting this up here, some of you will know that a few years back, I actually had a very severe episode of major depression. And because I have a familial history of Alzheimer's disease, and I also had a history of depression, I'm actually more likely and more susceptible to actually develop Alzheimer's disease of a particular type. So I'm interested in Alzheimer's disease now for very different reasons than when I was in grad school. And I'm hoping that some of you will be more interested in what you're studying for a variety of different types of reasons. So I'm hoping that your studies are not something that you're just taking for granted. So you're going to hear so many different things during your undergrad. And a lot of it is really just to finish, just as I did, what you were doing and what your course requirements are. And that's fine too. That's a really wonderful thing. But I also want you to understand that as an undergrad, because I've had a lot of undergrad trainees, that you can actually make a difference in terms of what you might be contributing to our field of knowledge, as well as directly. Nowadays, so many of you are making such a big impact at the level of the community, as well as outreach, that I think that many of you kind of underestimate what you are capable of doing. And I think that you can make an impact on different diseases like Alzheimer's or others. But I think that choice is always up to you. Just like I chose at the time to really insulate myself in my studies and not really pay too much attention to what was going on, that choice is yours. And that choice is always going to be yours throughout the rest of your life. So here's one of the reasons why we're kind of interested in knowing a little bit more about Alzheimer's disease. So here's his case study. So it's a 55-year-old male coming into St. Michael's emergency. It's the chief complaint with CC, as you see there, cognitive impairment, swollen ankles, abdominal distension, the computer tomography with the CT of the head and X-ray presents normally, so there's been no head trauma as far as you can see, and then a neurologist is brought in for a consult. And as a resident on call, what would you diagnose this individual with? So you have someone who's like their cognitive abilities are gone. They can't draw a clock normally, which is one of the things that they've been asked to do, and they've been asked to kind of go through things that most of us would take for granted. So they're incapable of drawing a normal clock, and when they're asked to do it several times, this is sort of what they come up with. So what's the problem with this individual? So again, like looking at all the stuff that you know, it's an older individual, it's a male, so more likely to have something like Alzheimer's disease. But then there are some other things. The cognitive impairment might make you think about Alzheimer's. But it turns out that many of these neurological conditions actually resemble Alzheimer's disease. And in fact, this individual was actually suffering from a stroke at the time. And the stroke was a result of the fact that they had a number of cardiovascular impairments that were already visible. Swollen ankles, meaning that, again, there was probably some vasculature issues, the stomach or abdominal distension, the same thing. So it's hard to make a good diagnosis, because this is the way that we diagnose Alzheimer's disease outside of just sort of sitting there and chatting with the individual. It's always done on pathology. And unfortunately, Alzheimer's disease right now, even though we've made great strides, and some of you will hear about Pittsburgh compound B or things about different types of radiotracers, we don't have a good way to diagnose Alzheimer's disease. So when we look at Alzheimer's disease, I'm going to talk to you a little bit about the gross pathology. I'm going to talk to you about beta amyloid, and that's where I want you to pay a little bit more attention. Potentially thinking about some treatment strategies related to the different kinds of enzymes that we have been able to identify, and then some of the genetics of Alzheimer's disease as well. And then this last link between cholesterol processing as well as late onset Alzheimer's disease. So again, we tend to be really lucky, maybe, we're not sure. So this was sort of a number of years ago from one of those earlier things that I posted. And a number of years ago, it was thought that with our aging population that we were going to see this really horrific rise and the sudden rise in Alzheimer's disease. And it could be either on the top part of the orange line, or it could have been anywhere on the bottom part of this orange line. And it turns out that as we get closer and closer to 2020, that we're lucky. It turns out that we had overestimated the amount of Alzheimer's. It's still climbing, it's still going up, but it turns out that we are not looking at the worst case scenario with these interventions related to exercise, things on diet, earlier diagnosis, better treatments, and more treatments on the way. It turns out that we're on the lower side of things. So it's not going to be quite as bad as we thought, but it is still increasing. And as our population gets older, that black line in the middle is probably the trajectory that we're kind of looking at. We also know that the mortality related to Alzheimer's disease is increasing. I'm not talking about the overall numbers. I'm talking about the deaths that have been related to Alzheimer's disease. So everything else is going down. So we have better treatments for things like HIV and AIDS. We have better things even for stroke. We're getting much better at it. But for Alzheimer's disease treatment and things related to Alzheimer's disease, we still are not very far along. So the mortality, the deaths that are related to Alzheimer's disease is actually still increasing. Many of you will know that Alzheimer's disease is a form of memory loss, but there's also attentional problems. And one of the systems that we'll learn about later, the one aspect that I'd like you to make sure that you know, the attentional system, when we look at it both in terms of psychology as well as in neuroscience, here's where we're looking at the acetylcholine receptors and the acetyl or cholinergic system. And we know that there are groups of cholinergic neurons that are lost and damaged in Alzheimer's disease that relate to this attentional problem. And as a result of all of these things and the areas of the brain that get affected, there's a lot of difficulty with planning as well as problem solving. There's a lot of confusion and spatial disorientation, especially as it starts to affect the temporal lobes and the hippocampi and the prefrontal cortex, where the neurons start to get lost as well. And of course, as my grandfather had some very, very severe and very pronounced mood changes, personality changes. Toward the end of his life, he literally was swearing at people all the time, like 100% of the time. No one could actually stand to be in the same room with him because he was so disagreeable to be around. So all of these things, again, suggesting that there's a big, massive change. Problem solving, that's one area of the brain. Memory, another area of the brain. Emotional control, yet another area of the brain. This is not one area, this is multi-areas within the brain. And then we have these behavioral things where we get a lot of confusion as well as disorientation around the time when the sun goes down, which is known as sundowning, that we don't really know if it affects some of the hypothalamic centers or not, but it's entirely possible that it does. Again, this is mainly for your own information because we're going to be talking about this in some more detail. When we look at the gross pathology of the brain, one of the things that we see is that the brain itself seems to be shrinking. The cortex tends to be shrinking. As the cortex shrinks, we actually see an increase in the size of the ventricles, the places that we normally see the cerebral spinal fluid. These actually enlarge as we lose tissue mass. The areas that get affected, again, the temporal lobe, which controls your memory, the prefrontal cortex, but almost every area of the brain will eventually be affected. These are the two major models that we think, because we can see this on microscopic pathology related to Alzheimer's disease. So we think, and we're not in 100% agreement, someday someone really, really smart will be able to finally convince everyone in the scientific community, whether it's beta amyloid that begins this process, or if it's hyperphosphorylation of tau that begins the process of Alzheimer's disease. But there is no consensus. It's split almost evenly, 50-50. Half the neuroscientists believe it's beta amyloid. The other half believe that it begins with hyperphosphorylated tau proteins, which should help to stabilize the microtubules. Again, in neuroscience, 50-50 is not very appetizing, and that's where we're at in 2018. We have no idea which of these two models is responsible for the loss of these groups of neurons. Again, if we're taking a look, there are specific groups of neurons that run throughout that get affected. So we definitely see widespread loss of neurons, and we see the atrophy. We think that there are some areas that are more important. So when we talk about the loss of memory, we're looking at the hippocampus, especially the temporal lobe as it gets affected. Then it will move through the association area, so it's spreading out from the hippocampus to the nearby areas of the cortex, known as the association cortices, and then it will start to affect sensory and motor areas, because we never talk about those. There are all sorts of problems with Alzheimer's. There's problems with sleep, there's problems with movement, there's problems with sensation. We just don't talk about them because the things that look most obvious to us are things related to memory or disorientation and or personality changes. We know that one area that seems to be important is the nucleus basalis of Maynard. So this NBM is an important one to make sure that you know. This is a cholinergic system. And again, some of you have taken PSL 300 before, so those are those cholinergic projection fibers that run throughout the rest of the brain. So they start off in a small group of cells, and then they spread out to other areas of the brain, and they're really important for things like attention. And if you lose these, you're not able to pay quite as much attention as before. It will also affect your sleep-wake cycles. It will also affect whether or not you are able to stay awake, etc. So again, here, going back to what I'm asking you to make sure that you know, and hopefully you had a little bit of time to review, because if you haven't done more than one lecture at this point, you should really be worried. I'd be worried. So if you're going back and thinking about this, if I'm specifically targeting cholinergic receptors, what gene might I be using or what promoter system might I use to actually target this nucleus basalis of Maynard, which is a cholinergic neuron? There are a number of possibilities. Just give me one of them. Do you remember what the synthetic enzyme is for acetylcholine? I could use acetylcholinesterase. There's a slightly better one than acetylcholinesterase. Yes? So I could use CHAT, or choline acetyltransferase, which is the synthetic enzyme, to target a gene that might be useful in delaying the onset of Alzheimer's disease. So again, we're looking at these loss of synapses, especially within these cholinergic projections. We are losing neurons through a number of processes, not just apoptosis, but there are other sorts of cell death pathways as well. We are seeing at the same time the accumulation of these beta amyloid plaques, which is not normally seen outside of the brain, outside of neurons. So this is outside of the neuron. And then inside of the neuron, we would actually see neurofibrillary tangles. The tau proteins are being hyperphosphorylated. There are too many phosphate groups on them. As neurons start to die, we will start to see astrocytic scars developing within the brain. And as it continues, we'll also see this immune response. So one of the theories that is predominant right now is the beta amyloid cascade theory. This is what starts the process of cell death and cell loss. There's a lot of good rationales for why beta amyloid. We see a lot of it. We see a lot of it outside of the cell. We see it in early stages of Alzheimer's disease. And these extracellular aggregates are not stuff that we would normally expect to see. And if we do this in culture, if we deliver these little groups of beta amyloid, which we call a plaque, it will actually cause cytotoxicity. So in a cell culture system, if I were to drop in beta amyloid, it would actually cause those neurons to start dying, and it's done through a number of different ways. It affects ionic concentrations, it affects the normal distribution of the different ions, etc. And these plaques are actually a combination of a cleaved form of amyloid precursor protein. So every single cell that you have, every neuron cell that you have in your brain actually already has amyloid precursor protein. The ones in your hippocampus have it, the ones that are in your medial temporal lobe have it, the ones in your prefrontal cortex, they all have APP. Neurons have APP. It's a normal integral membrane protein that we would normally expect to find. Just like we would expect to find deep sodium channels, we should expect to find APP within neurons. So again, it's something that's there, but something is going wrong with this process. And the reason why we think that this is a good candidate is that this APP that is normally occurring within your neurons can actually be processed by two different pathways. There is a non-amyloidogenic pathway, and this is where the amyloid precursor protein is cleaved by two different enzymes, alpha and gamma secretase. And then there's the one that causes beta amyloid to form. No surprise, this is where beta secretase
causing a beta amyloid fragment to occur is going to be predominant. So I'm going to tell you or ask you to make sure that you know this diagram for the final exam. So I can tell you in advance for the final exam that you should expect at least one multiple choice, and you might be encountering more than one, but also a potential short answer question related to APP processing and knowing the different steps that help to identify one pathway versus the other. So here's this integral membrane protein known as APP. And you don't have to worry that you're not going to be asked to draw out the structure of APP. But we have a large extracellular N-terminal fragment. Then we have segments that are embedded within the plasma membrane. And then we have this intracellular C-terminal tail. This is found in all neurons, all compartments of neurons. And here we have, endogenously, we have these different types of intracellular secretases. So in this non-amyloid pathway, this APP is going to be cleaved to form soluble APP alpha, and then as a result of this alpha secretase cutting, we get this larger fragment outside, the soluble APP alpha, and then we get this other domain that's inside called C83. C83 in turn, after alpha secretase is cutted, C83 is also susceptible to being cleaved by another enzyme known as gamma secretase. These are again endogenous enzymes that we find within our cells. And as a result of this cut, we will have what is known as P3, a smaller fragment here, and then another A-beta intracellular domain known as AICD. All of these, P3 and AICD, can be normally processed by the enzymes found within your cells and eliminated from your body. So this non-amyloid producing pathway is perfectly fine, and we think that this is a normal part of how this integral membrane protein is not involved in generating Alzheimer's. However, if, for example, something causes a shift, whether it's a genetic locus or whether it's something environmental or something causes the switch from this non-amyloid pathway to be imbalanced by this amyloid pathway, here is where we take exactly the same molecule, and now we are going to have beta secretase. Beta secretase cuts at a slightly different place on APP, so it's not going to produce APP alpha, it will produce a smaller variant known as APP beta. Again, nothing wrong with this soluble protein because it can easily be handled. However, one of the things that results is that this larger molecule that is left over can also be targeted by the same gamma secretase. This gamma secretase, though, now when it cuts, it results in the formation of amyloid beta. Typically, it's called amyloid beta 1 to 40, so amino acids 1 through 40, or A beta 1 through 42. So again, depending on where it cuts, it results in this A beta peptide being produced. This is what we think is toxic. So this beta secretase and then the gamma secretase working on the remaining C99 peptide is what causes the generation of this more toxic A beta or A beta 42. The reason why it's toxic, this can actually bind with itself. It can self-aggregate, and once it starts seeding and self-aggregating, it makes it easier for other A beta molecules to stick with it. Eventually, it forms an insoluble clump in the cell, and it becomes cytotoxic, as we talked about before. So again, this A beta pathway is a really important one. So in terms of neurofibrillary tangles, this is the other theory. And again, it's roughly split 50-50. Some scientists believe that this happens before A beta, and some people believe that the pathway that I just talked to you about happens first. And these neurofibrillary tangles might be the contributing factor that generates or precedes or actually is the mechanism by which neurons are lost in Alzheimer's disease. So these neurofibrillary tangles are intracellular, so they're found inside dying neurons. So these are composed of hyperphosphorylated tau proteins. So tau proteins normally bind to microtubules and stabilize these microtubules. However, due to overactive kinases, for reasons that we don't really understand all that well, when it gets phosphorylated by these kinases, tau will lose its affinity to bind to microtubules, and it will actually start to dissociate from the microtubules. These microtubules within these neurons start to disassemble, and then the normal axonal transport that is normally along these different microtubules is lost, and this leads to loss of neuronal integrity. These will also start to aggregate, and that's one of the sort of emerging theories in every single neurodegenerative condition that you'll learn about in your other third and fourth year courses. No matter what you're looking at, whether it's prion diseases, or whether you're looking at Parkinson's disease, or you're looking at any other neurodegenerative condition, in neurodegeneration now, in the modern sort of era, we're always looking at one seed molecule that is self-aggregating. So it could be beta amyloid, it could be hyperphosphorylated tau, it could be a prion protein, it could be something completely different, but there's always some sort of molecule that we think is accumulating aberrantly in neurodegeneration. And again, you don't need to worry about all of the diagram elements on here. This isn't something you should worry about for your final exam. But we think that as this accumulates within a cell, this will actually do two things. It disrupts the cell. It also puts a protein load that's toxic on the cell. The cell bursts open. So this happens first, and any of these sort of beta amyloid that's been accumulating will also be released at the same time. So this is why this precedes, for example, the beta amyloid, but we're not really sure if that is indeed the case. Maybe it's the other way. Maybe beta amyloid is affecting the normal ionic sort of concentration. Maybe the neurons are not properly being maintained, or trophic factors aren't being delivered, and so as a result, that neuron may start to degenerate its axons, causing this process to occur. So we don't know which one is actually correct. So these are some of the different possible treatment strategies. So if alpha secretase is important, maybe we could actually find a way to move it toward the non-amyloid generating pathway. So if we stimulate alpha, it will take away from the beta pathway. So they're kind of in balance. We want to move it toward the alpha pathway to get that CJH3 peptide and not the C99 peptide. Because gamma secretase is what actually processes the A beta, maybe there's a way that we can specifically inhibit the gamma secretase. There's been a lot of hope here at U of T as well. So if you're taking LNP courses, Janice Robertson and some other professors here have been looking at different immunization possibilities. So whether passively immunizing, which doesn't seem to work, with A beta antibodies, or actively immunizing so that the immune system of your own brain is going to start detecting these A beta earlier, might be a way to go. But these have also not been really successful in clinical trials that people have tried over the last 5 to 10 years. It's been, again, very frustrating in terms of the neurobiology that we haven't been able to really get a lot of luck with this whole immunotherapy, even though it's a huge field right now in terms of neuroscience. So if we believe in the A beta pathway, this will be sort of the dominant ways in which we can do this. Or if we're looking at tau being sort of the initiator, we might find ways or drugs to inhibit this tau aggregation. We might identify specifically the kinases that phosphorylate tau or hyperphosphorylate tau, and we might try to inhibit those kinases. We might actually work the other way. We might try to activate phosphatases to remove the different phosphate groups that might be impeding on them. And we might find different types of drugs to enhance microtubule stabilization, or we might find a way, very, very difficult and very challenging, to increase tau clearance. What would be really challenging with this particular strategy that scientists originally thought of doing 15 years ago, but actually didn't work very well? Any ideas? What makes that tough?
Because tau is responsible for the microtubule stabilization, and you would actually do more damage if you use tau.
And it's an intracellular protein. It's normally there for stabilization. How do you get the ones that are associated with microtubules versus the ones that are falling off the microtubules? Very, very challenging. And removing something from an intracellular space in and of itself is also really challenging. So we're kind of stuck. We don't really have a good way right now to be able to have different therapies. The immunization methods that are designed against A beta pathways, they're sort of the dominant sort of approaches therapeutically right now. But again, we have no way of knowing if that theory is actually correct, if that's sort of the initiator or not. Yes?
Do one of these problems cause, for example, do alpha beta plaques cause phosphorylates causes alpha beta plaques?
So again, there are two different theories and two different camps. What initiates this and what follows. You see both. So there's no doubt that these are both involved, but we don't know what causes, if it's A before B or B before A, we don't know.
But could they cause each other though? Like, it doesn't matter or could they cause each other?
So originally, in the 1980s, when scientists were first able to biochemically extract and isolate the A beta peptides, there was a lot of hope that it was actually A beta because when you add it to cultures, like normal neurons, they start degenerating. So that was sort of, it's found extracellularly to begin with, and if it's found and you accumulate more of it in the extracellular space and it's killing neurons, that was sort of the hope for what A beta is, but there's no clear evidence that that actually initiates all of the downstream cascades that also activate tau kinases. Okay, so again, as I sort of wrap things up here, here's some of the difficulties with the genetics. So I just told you a whole bunch of things related to why I think there's a strong genetic component in my family. So there's a lot of familial history. Once I started asking, because I don't know how many of you actually talk to other members of your family. So my kid comes home and then she goes upstairs and then she texts me when she needs something. So we don't really talk, even though we're in the same house at the same time. And I don't know how much you talk with everyone in your family, but it looks like there's a very strong sort of family history for me. But when you look at it overall, the familial inherited forms of Alzheimer's disease, they are very, very rare. We know most of the genetic variants. Of all the cases of Alzheimer's disease, only 5% of them are actually, or can be attributed right now to a genetic problem. And we know what they are, presenilins 1 and 2, or a problem with like having too much beta amyloid, but we know what the genetic causes are, but it's only 5%. That means that 95% of cases for Alzheimer's disease are sporadic. We just don't know what the cause is. Could it be something in the environment? Could it be eating too many grain cookies? Could it be having too much coffee or not enough coffee? We just don't know. And so when we look at this, this becomes really interesting. So one of the things that we do know when we look at Alzheimer's disease, the familial form, the one that gets inherited, it becomes much more severe. So it starts at around ages 40 to 50 years of age. So the sporadic cases are usually later in onset. So when you hit the age of 65 and over, that tends to be much more common for the sporadic form. And the symptoms, though, are identical. The areas of the brain that get affected, though, are identical. And when we look, so far, the vast majority that we look at, about more than 50%, to be quite honest, can be explained by known mutations in genes that encode for APP as well as presenilin. Unfortunately, I'm not going to be able to get through presenilin today, but I will tell you a little bit about some of the other mutations. So APP mutations, generally speaking, are relatively rare. The average onset of Alzheimer's disease in individuals with APP mutations is about is in their early 50s. And again, one of the reasons why we know this is that if you were to look at someone who has Down syndrome, in the Down syndrome, they will actually have an extra copy of chromosome 21. And on chromosome 21, we carry the APP gene. So they have like 3 copies of APP. They're overexpressing APP. And as a result of that increased load in APP, they're just generating much more of the A beta. So again, one of the things that we also know that when we identify these mutations that alter this APP processing, it's usually in that pathway that we looked at earlier. There's an increase in the beta secretase pathway. There's an increase in the two different forms. And this is one to maybe highlight for potential multiple choice test questions. When you look at the different fragments, you can actually form A beta 1 to 40 or A beta 1 to 42. The A beta 1 to 42 is the more toxic form. And in cases where you have or where you have cases that involve APP mutations, they have much more of the A beta 42, so it tends to be a little bit more toxic for them. And these tend to form more fibrils. So again, there's a lot of evidence to suggest that there are some genetic mutations. And the pre-stimulants, again, just to give you a quick overview, you can't get away from pre-stimulants because the prof who was just down the hall and on the first floor of the TANS building, or this Center for Research in Neurodegenerative Diseases, when I got started, actually is the Canadian scientist Peter St. George Hislop that identified pre-signaling 1 and 2. And he was the first individual ever to be able to describe a genetic locus for why there is Alzheimer's disease. And what he found was that these pre-stimulants are mutated in individuals that have Alzheimer's that is due to a genetic cause. So these pre-stimulants, this is the reason why these pre-stimulants are important. Pre-stimulants are part of this complex. So there's this gamma secretase that I told you before. This gamma secretase itself, the one that actually cuts either the C99 or the C83 peptide, it consists of a pre-signalant, 1 or 2, another molecule known as nicastrin, APH, and then PEN2, and all of it kind of looks like this. The pre-signalant is the thing that actually has proteolytic activity. It's the thing that actually cuts. So when we talk about gamma secretase, we're really talking about pre-signalant 1 or pre-signalant 2. If you have a mutation that affects the enzymatic processing by pre-signalants, it actually lowers the age of onset. So the more mutations you have in pre-signalants, the lower the age that you're going to have in terms of having Alzheimer's disease. The vast majority of pre-signalant mutations are in pre-signalant 1 rather than in pre-signalant 2. And again, the same thing that we saw with APP, if we have more of the A beta 42, which happens if you increase the mutations in pre-signalant 1, so the enzyme is much more active, it will actually cut more in favor of the A beta 42. So again, just to wrap things up a little bit in terms of Alzheimer's disease, the thing that you've probably heard the most of in terms of risk factors for Alzheimer's disease outside of pre-signalants or outside of things that get inherited are things like ApoE. And I'm sure you've heard of ApoE before. So these are not directly responsible for disease onset. And some of these different genes that are associated with apolipoprotein E or ApoE has been identified as the largest risk predictor of late onset. So that means when you're over the age of 65, not the familial form, but if you have one of these different variants, it's actually a risk predictor that you will develop late onset Alzheimer's disease. So ApoE is normally formed by the microglia in the brain, as well as in the astrocytes within the brain. So ApoE, its major function once it gets released by these microglia and astrocytes is to solubilize cholesterol within the brain and allows for it to be transported. We don't only have ApoE, by the way, in the brain, we have it in a number of other tissues within the body. There are 3 of these different alleles that have been associated with apolipoprotein E, ApoE2, ApoE3, and ApoE4. So if you were to take a look, ApoE4 is the one that actually gives you the largest risk of developing Alzheimer's disease later in life. And it's this simple switch in these 2 amino acids. So if you have 2 arginine residues, you are at an increased risk of developing Alzheimer's disease. And it turns out if you have 2 cysteine residues, you're actually protected from developing Alzheimer's disease in terms of the late onset. So which of these 2 would you rather have? ApoE2 or ApoE4? Or none of the above? it actually puts you at an increased risk of dying of a heart attack. So you either die from Alzheimer's disease or a heart attack. So here, as in life, it's best to just be normal. Just be normal. So having ApoE3 is actually the best choice and the best scenario that you can have. So ApoE2, although it's protective against Alzheimer's disease or an increased risk of cardiovascular disease, and ApoE4 is putting you at higher risk. So actually just being normal and having ApoE3 is the best way to go. Okay, so again, ApoE4 normally binds to A-beta, helps to facilitate its clearance, but ApoE4 clears A-beta the lowest out of all of the different isoforms, which is why we don't think it's very useful for you. It also gets lipidated at the lowest level as well. And again, we won't go through all of the details here. So I think that's sort of it for what I wanted to go through with you. So I'm just going to wrap things up like really quickly. If you guys can stay for a quick picture, that'd be great. And I just wanted to share a couple of things with you. So I'm hoping that down the road, so you guys are finishing off summer school, which again, it's been a really tough grind for a lot of you. Some of you are working, also going to summer school. Some of you are sick, going to summer school. Some of you are sick on summer school. And so it's been a really rough grind. So I know that we're getting to the end. But I also wanted to tell you as you're going into your senior level years of study, I want you to maybe think about how you're going to change things. So I'm hoping that you've changed things or been changed by your sort of experience here in class, and I'm hoping that it will continue on, that you've met more people that you didn't know before class and you know more of them now at the end of class, or that you learned a little bit more than what you knew before class. And I'm hoping that you'll do the same. Wherever you're going next, that you're going to change and impact whoever you get in contact with down the road. So I don't think that any of you in 3rd or 4th year should ever feel like you can't make a difference, whether it's here at U of T or after you graduate. Because I certainly felt at the time when I was in undergrad that I shouldn't actually have those types of goals. But I don't want you to have that sort of lowered expectation. I want you to be able to change things that are going around in the environment around you. So I want you to, again, think about this. So we're about 1 week, exactly 1 week. 1 week and I think 4 hours, is that right? 4 hours away from your final exam. So let me put this out to you as well. So there is a quiz this week. This is the last one. Some of you, again, like to play really, really dangerous, kind of waiting until like the last half hour. Not sure if you get like a little dopamine rush, you get this little spike, and you really enjoy it. If you do, great, you'll have one last chance at it. I would really recommend not doing that, but that's the choice. If you have missed one for whatever reason, so you were sick, or you just forgot about the time, or whatever the reasons were, you have until this Thursday to let me know, and then we'll try to work out an arrangement of kind of reshuffling things. Again, just to make sure that you're all aware, so the final exam is not cumulative. There's a lot of material. These last 4 lectures and today's lecture, which is part of that 4, there's a lot of material that you need to stay on top of. The short answer questions, out of the 4 that you get, 2 of them are very heavily applications based. Some of them involve being able to draw, not quite like this, but more like this on your final exam. So being able to tell me that you understand how to design a construct. So that happens twice on your short answer questions. So it's really hard for you to kind of make sure that you go through and study that pink sheet that I gave you earlier. Kind of take you through the steps that you'll have to know how to do that. Please keep track of these different deadlines. How many of you would like one of the multiple choice test questions in advance? Okay, so I'm going to tell you, because not all of you are logging in every single day or even like once a week. I'm going to tell you that I'm going to be posting that one question and maybe one of the short answer, parts of one of the short answer questions in advance on the announcements page. It's only going to be available on Friday, and it's only going to be available for 6 hours, and then it will disappear. Friday. Okay, if I'm working on Friday, Guess what? You're working on Friday too. Friday doesn't work for all of you? No? Shall I just leave it on there for you? Okay. I will leave it on there for you. So I want you to be checking on the announcements page. I mean, some of you literally log on like once or twice a week. That's not enough. And make sure that you're paying close attention to the announcements, because there might be useful information for you on the final exam related to what do I need to know from that slide on the exosomes, because that will probably also be an announcement for all of you as well. So please make sure that you're checking. So also check up on court case. If there's anything missing from any of your marks, let us know in advance. It's a lot easier to fix those before your marks get submitted than to try to make a grade amendment. So I will, as I mentioned to you before, give you at least one of your multiple choice test questions in advance. Give you a really good idea of what to expect on the short answer. That will be posted directly on there for you on the announcements page. Last thing I'm going to mention before we formally wrap up the course is that if you need extra help outside of Friday, because no one apparently likes Friday, you can come and see me. There is a tutorial group that has already met twice on Tuesdays and Thursdays, and we have another set that's going this week. The size of the classroom is a little small, but will accommodate based on how many of you are interested. So just let me know, because I don't want you to go through the final exam, because the worst feeling that you've ever had, because I've had it like 5 times in my career here at U of T. So I had it in physical chemistry, I had it in organic chemistry, I've had it in physiology, and I've had it in LMP twice. You ever open up a test booklet and then you look at the question and you have no idea what it's actually asking you? I've had that, and I don't want you to have that on the final exam. So I want you to have, you know, a proper preparation for it. Just let me know how I can help you with that. And I think I would just like to leave off by just saying that you were a really brilliant class. I'm sorry that I didn't get to know all of you all that well. I hope that we can continue to stay in communications. And I just wanted to thank you for being such a brilliant class. If you can, please stay for a quick picture, help yourself to some food. We're just going to wrap up here. I think we're going to try to take a picture outside, and it'd be really great if you could stay for that group picture. Follow Max, so I'll tell you where to go and stand outside. All right, thanks everyone. I'll see you all later on.
So if you guys want to be part of the